Welcome back to Dr. Liu's VCE Biology. Today's topic is photosynthesis, cellular respiration, and biotechnological applications, as presented in the 2024 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. A diagram of an animal cell is shown here. Location A in the cytoplasm may contain the enzymes for glycolysis, as this process occurs in the cytoplasm. Location B is the endoplasmic reticulum, which does not participate in the Krebs cycle. Location C is the cytoplasmic membrane, which is not the site of protein synthesis. Location D is the mitochondria, which is where aerobic respiration takes place, not anaerobic respiration. NADH and FADH are electron carriers, or energy transfers. Their main job is to carry high-energy electrons from earlier stages of cellular respiration, like glycolysis, and the Krebs cycle, to the electron transport chain. The energy they are carrying is stored within the electrons. They don't provide energy in a usable form directly to the cell. Instead, they donate these electrons, which fuel the process of ATP production. ATP is the cell's energy currency, or energy carrier. ATP is the molecule that directly provides energy for most cellular processes. The energy is stored in the bonds between its phosphate groups. When a cell needs energy, it breaks a phosphate bond, converting ATP to ADP, adenosine diphosphate, and releasing energy. Because it holds and carries this readily usable energy, ATP is considered the final energy carrier. At the same time, NADH and FADH are temporary carriers that shuttle energy to the final step of production. A scientist growing cells in a culture noticed a baked bread smell, and subsequent chemical analysis of the tissue culture medium detected ethanol. This finding indicates that the cells were performing alcoholic fermentation, a form of anaerobic respiration that produces ethanol. This process is characteristic of yeast, which might be a contaminant in the cell culture. Farmers often use greenhouses in cold climates to speed up crop growth. The greenhouse's ability to trap heat elevates the internal temperature, which in turn increases the rate of photosynthesis. However, a greenhouse does not change a plant's chlorophyll concentration, oxygen levels, or pH value. In the light-independent reactions of photosynthesis, the energy from ATP and electrons from NADPH are used to convert carbon dioxide into glucose. Based on the graph, the total amount of oxygen is the sum of the oxygen released and the oxygen used. At 16 hours, this total value is calculated by adding the 200 units of oxygen released to the 50 units of oxygen used. If light intensity decreases from bright to dim, the rate of oxygen released by plants will also decrease. This is because a lower light intensity slows down the light-dependent reactions of photosynthesis. Results were repeatable means that consistent results were obtained when the experiment was performed multiple times under identical conditions. To ensure the results are truly repeatable, the experiment should be run again, with the same sequence of 12 hours of darkness followed by 12 hours of bright light. The investigation examines how different sources affect ethanol production. In this experiment, the independent variable is the type of source, and the dependent variable is the yield of ethanol. Variables such as yeast concentration, temperature, and oxygen levels are not being investigated. Therefore, option B, which describes the relationship between algae and ethanol yield, is the correct choice. A scientist investigated the effect of rosiglitazone on glucose metabolism in actively growing human liver cells. Over 24 hours, cells in 2 Petri dishes were grown under identical conditions, with one dish receiving 5 micromoles of rosiglitazone, while the other served as a control. Rosiglitazone is known to inhibit the uptake of pyruvate into the mitochondria. Since rosiglitazone inhibits pyruvate's entry into the mitochondria, the cell must increase its glucose consumption to meet its energy demands. This reliance on fermentation for energy also causes a rise in lactate production. In cells without rosiglitazone, pyruvate can enter the mitochondria. This allows the cell to obtain energy through aerobic respiration, where NADH and FADH from the Krebs cycle transfer their electrons to the electron transport chain on the inner mitochondrial membrane, cristae. An experiment to test whether 2-deoxy-D-glucose, 2-DG, is a competitive or non-competitive inhibitor of hexokinase would involve a series of test tubes, all with the same amount of 2-DG, and liver cells under identical conditions. The key variable would be the glucose concentration, with a control tube containing no glucose. If 2-DG is a competitive inhibitor, a high concentration of glucose will eventually outcompete it for the enzyme's active site. As a result, the rate of glucose 6-phosphate formation will increase and eventually approach the rate of the uninhibited enzyme. If 2-DG is a non-competitive inhibitor, the inhibition cannot be overcome by increasing the glucose concentration. A student designed an experiment to determine how different colored lights impact plant growth. The setup involved placing identical seedlings into individual glass containers and monitoring their gas and water levels. A key part of the procedure was keeping the seedlings in darkness for 2 days before the experiment started to ensure they had used up any stored energy. The data shows that the group of plants exposed to green light did not grow, as well as those under other types of light. This is because chlorophyll primarily absorbs red and blue light, while it reflects green light. The green filter blocks the light the plant needs most, which results in a lower production of ATP and NADPH in the light-dependent reactions, thus slowing growth. Genetic engineering could be used to improve seedling growth by introducing new genes. These genes would code for accessory pigments, enabling the plants to absorb wavelengths of light that chlorophyll typically does not. By absorbing a wider spectrum of light, especially the green light that is often reflected, the plants could capture more energy and grow more efficiently. In an experiment with varying carbon dioxide concentrations, seedlings from C3, C4, and CAM plants were exposed to white light. After 7 days, the group with the least growth was the one exposed to low CO. This plant is likely a C3 plant, as C3 plants are highly susceptible to photorespiration. Under these conditions, oxygen competes with carbon dioxide to bind to the enzyme rubisco, which reduces the efficiency of the light-independent reactions and leads to less glucose production. This section is worth 25 marks, which contributes 20.8% of the total 120 marks. It covers a range of topics in biology, including photosynthesis, cellular respiration, and biotechnology. Multiple choice questions, 9 marks. This part of the exam focuses on core biological concepts, cellular processes, Questions 6, 8, and 11 cover the functions of different cell locations and the roles of molecules like NADH and FADH in energy transfer. Metabolism and environment. Questions 9 and 15 examine conditions that lead to ethanol production in cells and the production of biofuels, while questions 10, 12, 13, and 14 explore how environmental factors such as light intensity and temperature affect photosynthesis and plant growth. Short answer questions, 16 marks. This section requires more detailed and analytical answers. Experimentation. Question 3 delves into specific experiments, such as the effect of rosiglitazone on glucose metabolism and how to identify a competitive or non-competitive inhibitor. Applications. Question 4 covers practical applications of these concepts, including the impact of different light colors on plant growth and the use of genetic engineering to enhance plant growth. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.